So welcome to today's lesson on the uses of radiation. So there's only one learning objective for today. Describe the uses of radioactivity in both medical and non-medical traces, also in chemotherapy and in radioactive dating. So in terms of using it in medicine, they have various uses. That includes imaging, chemotherapy, and the use of traces, which we'll look at in more detail soon. And the radioactive isotope that you choose depends on two things: its half-life and the type of radiation it gives out. So let's first look at medical traces. Okay, so traces are essentially used to investigate what's inside the patient's body, if there's any blockages, for example. And this is the reason why we use gamma emitters and beta emitters, because if we use alpha particles, they can't escape the body because the body's skin and fat layers and whatnot is too thick. So an example here we have is technetium-90. It has a half-life of 60 hours. That half-life is reasonably short, so it means it decays within the body, because if it's too long, then you end up causing cancer, because the half-life is just too long. It exists in the body too long, basically. And it decays also into a stable isotope, which is also important, because if it decayed into an unstable isotope, then you would still have radiation emitting from the person inside them, and that would be quite bad in terms of giving them, say, cancer. So how does it work? Okay, so the example of gamma radiation. So if the radioisotope emits gamma particles, or gamma rays, then it means that those gamma rays can be detected, and you can kind of triangulate where there could be blockages or brain tumors, for example. This is why we use gamma emitting radioisotopes like technetium. Okay, once the molecule is fixed to, for example, a target molecule, so you fix your radioisotope to a target molecule which targets a particular area of the body, for example, the pituitary gland, if you maybe have cancer in that or want to know that you have, and then you can detect if there's any irregularities because it's going to emit from that particular position, and you can detect the gamma rays. Okay, then we have, say, the gamma knife. Okay, so that's basically giving a very high radiation dose of gamma radiation, but small enough so it doesn't really affect the surrounding tissue. So you're basically focusing on, say, a brain tumor. You fire the gamma rays at that brain tumor. It destroys the brain tumor, but it doesn't destroy the surrounding healthy cells. And an example of a very good source for gamma rays for this is the cobalt-60 isotope, because again it has a reasonably short half-life, but you don't really have to worry about half-lives, because remember the cobalt-60 will be outside the body in this particular case. You're firing the gamma rays into the body. Then we have chemoradiotherapy. Okay, so what we're doing there is we're firing high doses of gamma rays that can focus on cancerous cells in particular. Okay, so it can be done from outside the body, okay, firing X-rays and gamma rays into the body from outside. Again, cobalt-60 is a good source for this. Or that you can actually place a target molecule, which targets, for example, if you have cancer in the prostate, then the molecule will target that particular prostate organ, and it means it can be released within the prostate. So you have a target molecule and the radionucleotide, which will emit gamma rays and kill at the source. And again, the half-life is about 5.7 years, because most of this is done outside the body. And it's also relatively stable, the isotope, because it doesn't decompose into a more unstable isotope. Then we have sterilization, which is not a medical use, this is more industrial uses now. Okay, so sterilization is basically firing gamma rays at particularly medical equipment. Okay, and also the advantage of that is medical equipment can be in their packages, so that increases the sterility. Okay, and also food can be hit with gamma rays, because it can kill microbes and fungi that are growing on the food, making it unedible. And gamma rays can be used to kill those particular beasties before you eat them, which is an advantage. And then we have tracing leaks. Okay, so this is using beta particles essentially. So you can add a radioisotope which emits beta particles to a fluid, and that means that when the leak comes along, you can detect where the leak is, because that's where the concentration of beta particles will be the largest. Okay, the largest. And then we have automatic thickness monitoring. Okay, so you can actually detect like the thickness of paper using, again, beta emitters. So what happens is that as the thickness of the paper increases, you have a detector on one side of the paper and a beta particle emitter on the other side, and as the thickness increases, what happens is the beta particles can't get through. There's now no longer a signal which says to the industry, the paper, say the paper milling industry, that the paper is now too thick, you need to stop the process and see if there's a mistake in your presses, and then you can start up again. It's only when the thickness is set that the beta particles can get through and be detected. Then we have, so this is basically the properties of this particular source. It has to be a beta emitter because basically it's a pass through the paper. And gamma rays would be useless because they just shoot through no matter what the thickness was. Alpha particles wouldn't work because they wouldn't get through the paper. And it has to have a long half-life source in order to get reduce the possibility of false positives. So the isotope suggested is strontium-90, which is the beta emitter with a half-life of 29 years. For smoke detectors, what happens is that obviously alpha particles are quite effective for this, because what happens is the alpha particles can't get from one side of the circuit to the other. If there are smoke particles present, they hit the smoke particles and they get stopped. Okay, and that means that no current flows, and this current drop sets off your fire alarm. It'll beep, beep, beep. In terms of the properties required for the source, has to be an alpha emitter with a long half-life, because beta and gamma will just shoot through the ionizing particles, and that means you'd still have a current and you wouldn't know there's a fire going on. So the isotope commonly used is americium-241, which is an alpha emitter with a very long half-life relative to humans of 433 years. Another use is radioactive carbon dating. Okay, so as we previously learned, living material contains a proportion of carbon-14, and this isotope is produced basically when high-speed neutrons hit the nitrogen gas within our atmosphere. And this is pretty much how the system actually works. So what you have is you have the sun emitting these high-speed neutrons, and what that does is it obviously creates carbon-14 in the atmosphere. And when this carbon-14 is absorbed by plants through the process of photosynthesis, it means any living material will contain, including you, a certain proportion of carbon-14 when you eat the plants. Okay, so as soon as you eat the plants, you also get this carbon-14. Obviously, when you die, you can no longer absorb because you're no longer eating, and plants can no longer expire carbon dioxide through the process of respiration, and they can no longer absorb carbon dioxide through the process of photosynthesis. So that means the amount of radioactive carbon-14 starts to decrease as soon as you die. And the age of the living material that's now dead can be estimated by comparing it with a residual radioactive carbon content with living material. Okay, so that's what you see here. The turkey eats the plant, and then when it dies, it's no longer emitting carbon dioxide or absorbing it. So you can then detect that against certain standards. There are limitations, of course. So it assumes that cosmic radiation levels have been constant all through the history of the Earth, okay, because that means carbon-14 levels have been constant. You have to correct the carbon-14 levels based on objects that no longer have living material, like Egyptian pharaohs, for example. So we know when they were around and we can detect that, use that as a standard, as it were. And it's only limited to dead things which have been dead for about 60,000 years. Otherwise, it gets quite inaccurate. Okay, and the final thing to look at is dating rocks. So there's a number of techniques used, but one example is looking at uranium-238 decay. So it decays basically all the way down into lead through thorium-234 until lead-206 is formed. These are called the nucleotides. And older rock samples will have a greater amount of lead content. So you can determine the ratio between uranium-238 to lead to determine the age of the rock depending on or comparing two standards. So that's the end of quite a long lesson.